Hello everyone, I'm Anas Al-Ubaidi and this is the fourth session or fourth lecture within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program. And this chapter is one of the chapters from the curriculum of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. And the chapter of 24 is about conceptual and theoretical monitoring, while the topic of this session, session 4, is about monitoring of drug-being and drug-non-being interaction trials. Introduction. We'll start with the definition. As drug-being interaction refers to how two or more drugs react with each other when co-administered, potentially altering efficacy or safety. Drug-non-being interaction involves the effect of external factors such as food, alcohol, or smoking habits on drug absorption, distribution, metabolism, or elimination. And ADME as ADME, absorption, distribution, metabolism, or elimination, or excretion, the same. And here is the pharmacokinetics. So the drug-non-being interaction involves the effect of external factors, as for examples, not limited to food, alcohol, or smoking habits, on the drug pharmacokinetics. Part of it, or all the whole pharmacokinetic, or part of it, only absorption or distribution or metabolism or excretion or elimination. Significance in clinical research prevents harmful interactions by predefining safety measures, optimizes therapeutic regimens for target populations, guides prescribing patterns and labeling. As for example, here a trial studying the interaction between anticoagulants and non-steroidal anti-inflammatory drugs evaluates increased bleeding risk. Similarly, a drug-non-being trial may explore whether grapefruit juice alters the metabolism of statins. As the first one is the drug-being interaction, and the second part here of the grapefruit juice example is the drug-non-being interaction. Key points. We'll start with the key components of monitoring drug-being and drug-non-being interaction trials. Safety monitoring. Purpose. Safety monitoring ensures the identification and timely management of ADRS or toxicities that may result from interactions. ADRS can range from mild symptoms to life-threatening conditions. How it's done? Baseline lab testing followed by periodic assessments, as for example, liver function tests for hepatotoxicity or renal panels for liver toxicity. Monitoring and recording of cardiac parameters, as for example, QT prolongation with antiarrhythmics, identifying interaction effects that amplify toxicity, as for example, combined anticoagulants and non-steroidal anti-inflammatory drugs, NSAIDs, increasing bleeding risk. And here is a summarized diagram unveiling safety monitoring dimensions. As safety monitoring, we have baseline lab testing, cardiac parameter monitoring, and interaction effect identification. As for example, for safety monitoring, patients co-administered an ACE, angiotensin converting enzyme inhibitor, ACE inhibitor, and non-steroidal anti-inflammatory drug, NSAID, by show increased creatinine levels. As safety monitoring protocols include regular renal function surveillance. As here, that's why clinically it is not suggested, not advised, and not recommended to give same patient with multiple medications or prescriptions, not to prescribe or not allow any patient, even over the counter, to take ACE inhibitor as it is prescribed, and non-steroidal anti-inflammatory drug, whether prescribed or over the counter, not to be given together for the same patient, especially for long term use of both together. That's why one of the problems or complications or medical consequences would be increased creatinine levels, and by that meaning renal or kidney dysfunction. Safety monitoring protocols include that's why here include regular renal function surveillance if we have a patient or even a participant taking both. B. After safety monitoring, efficacy assessment purpose to evaluate if an interaction improves or diminishes the effectiveness of the drugs involved. This step can identify synergistic combinations or uncover drugs that cancel out each other's effects. How it's done, clinical endpoints, as for example, target blood pressure for antihypertensive combinations, assessment of symptom resolution in infectious disease trials, as for example, antibiotic steroid interaction or interactions. As for example, here a trial combining an oral antibiotic with a proton pump inhibitor, PPI, determined the PPI reduced antibiotic bioavailability, rendering therapy ineffective. This is only an example. Tracking. Purpose: patient adherence is critical, but especially in interaction trials, since suboptimal compliance can distort findings. Poor adherence may lead to underestimated risks or overlooked beneficial synergies. How it's done: pill counts, digital tracking devices, pharmacokinetic drug level measurements, or patient diaries. Newer innovations: wearable sensors to track the timing and dose of administered medications. As for example, for antiretroviral therapy (ART), adherence and drug-drug interaction studies, discrepancies between patient logs and measured drug levels are red flags requiring resolution. Steps to effective monitoring. Step 1: protocol development. Develop a well-defined study protocol. Clearly specify objectives and endpoints focused on interaction effects. Include pharmacokinetic, as for example, drug concentration curves, and pharmacodynamic parameters, for example, additive therapeutic effects or increased toxicity. Enrich patient inclusion criteria to standardize confounding factors, as for example, restricting participants with incompatible comorbidities. As for example here, a diabetes monitoring trial testing a novel hypoglycemic drug in combination with existing insulin therapy must define hypoglycemia incidence as one safety marker. And here's a summarized diagram on protocol development for monitoring, as we have standardized patient criteria. We'll start with example application. Standardized patient criteria include pharmacodynamic parameter, include pharmacokinetic parameters, and then define objectives. Step 2 here, data collection. After protocol adherence, we have, or protocol development, we have data collection. Accurate data collection is crucial to draw meaningful conclusions. Tools like electronic data capture (EDC) systems simplify recording and standardization. Patient records, laboratory data, and real-time monitoring are typically part of data acquisition. As for example, during trials evaluating anticoagulants and antibiotics interaction, irregularly collected INR values provide evidence on drug interaction impacts on blood clotting. Adverse event AE escalation. A streamlined AE escalation pathway ensures quick actions when moderate or severe issues arise. Utilizing standard terminology such as MID-DRA or CTC-AE simplifies documentation and communication between teams. Example: elevated ALT/AST enzymes during a hepatotoxic interaction trial immediately trigger suspension of drug administration or for involved participants. Step 4: interim reviews and safety meetings. Regular periodic reviews by a Data Safety Monitoring Board (DSMB) strengthens safety oversight. As we know, this board is one of the boards that work independently in order to avoid bias, avoid errors, and avoid any influence. DSMB is as one example. So Data Safety Monitoring Board. Halt or pause studies showing alarming trends, modify protocols when needed to protect participants or patients. As for example, a psychotropic drug trial is paused after DSMB reviews find drug interaction with another compound raise sedation beyond safe thresholds. Step 5 final data analysis and reporting. Advanced statistical methodologies quantify interactions, linear and nonlinear regression models, or survival curves like Kaplan-Meier plots are often applied, as for example, graphs may show grapefruit juice nearly doubles the plasma concentration of certain drugs metabolized by CYP3A4. We have here tools and techniques for monitoring advanced EDC systems, electronic data capture platforms like Meditate Streamline, one example, other examples like ClinTrack or Inform as well. Streamline data entry, real-time anomaly detection and reporting critical for large-scale clinical trials. Example, flagging trends in lab abnormalities such as high potassium levels in patients on spironolactone and ACE inhibitors. Here is a summarized diagram on advanced EDC system workflow, as we have data entry, real-time anomaly detection, anomaly detected. We have here continual monitoring. Here we have flag trend, after flag trend report regeneration, and then clinical decision making. Pharmacovigilance PV databases after advanced EDC systems, databases like Vigibase allow post-marketing trend analysis. Real-world population data often reveal low-frequency interactions and detected in trials. As for example, identifying that dizziness increases among patients combining two antihypertensives, requiring customer labeling adjustments. Modern statistical methods, from regression analysis to simulation software, statistical tools ensure results are scrutinized and accurately interpreted for precision medicine applications. We have biomarker tracking, monitoring biomarkers for safety, efficacy, or target interaction endpoints such as inflammatory markers, CRP, or liver enzymes, AST/ALT. As for example, plasma glucose levels in trials, adding a second glucose-lowering medication for diabetes management. Drug interaction checkers, real-time drug interaction software like Lexicomp significantly aids trial planning. As for example, discovering a statin interferes with a trial drug can spare months of wasted research focus. Summary tables. Next, we have tables to summarize what we have covered in this lecture. Table on definitions and significance, the aspect, and then we have a drug-being interaction and drug-non-being interaction. As here, definition for drug-being interaction examines effects when drugs are co-administered, altering efficacy or toxicity. Drug-non-being interaction, a definition studies how external factors like food, alcohol, or smoking affect drug behavior. We have the focus, significance, clinical application, and example. And for each drug-being interaction and drug-non-being interaction. We have table on key components of monitoring. Component, purpose, activity example. Components, we have safety monitoring, efficacy tracking, compliance monitoring, data collection, and real-world evaluation. Table on steps to effective monitoring. Step, objective, activity, outcome example. Steps, we have protocol development, data collection, AE reporting, review and adjustment, and then data analysis. Tools and techniques for monitoring. Tool/technique, purpose, functionality, benefit example. Tools, techniques, we have electronic data capture, EDC, PV systems, statistical analysis tools, interaction checkers, and biomarker evaluation. We have a table on examples of successful monitoring, scenario, challenge phase, monitoring action taken, outcome achieved. Scenarios, we have drug-drug interaction, drug-food interaction, polypharmacy. As drug-drug interaction is drug-being interaction, drug-non-being interaction, drug-food interaction. Polypharmacy, meaning more than average of 5 per some resources of prescribed medications, meaning a long list of prescribed medications, adherence gaps, and outlier identification. Application table, scenarios. For each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios, we have monitoring bleeding risks in trials combining anticoagulants and nonsteroidal anti-inflammatory drugs, evaluating statin metabolism affected by grapefruit juice, identifying QT prolongation through or via ECG or EKG risk, in antiarrhythmic co-administration, measuring blood pressure during combined antihypertensive drug efficacy testing, tracking adherence to ART and proton pump inhibitor regimen trials, adjusting doses in elderly participants, showing renal impairment in polypharmacy trials, addressing reduced contraceptive efficacy in trials combining antibiotics and birth control, evaluating changes in liver enzymes due to herb drug interactions, as for example, St. John's wort. We have resolving severe ADRS flagged during CNS drug and trials involving CYP 450 enzyme inhibitors, employing EDC electronic data capture systems to flag high lab value outliers during interaction trials, studying bioavailability changes when administering lipophilic drugs with high fat diets. We have issuing site safety alerts for managing high bleeding frequency in anticoagulant trials, improving adherence to standard blood sampling protocols in warfarin antifungal interaction studies, investigating GI gastrointestinal adverse events and studies assessing antihypertensive combinations affecting elderly patients. Next, we'll be revising what we have covered through this lecture through some via some multiple choice questions or MCQs. Question 1. What is the primary focus of drug-being interaction trials? A, to study environmental effects on drug metabolism. B, to test drug combinations for potential reaction changes in efficacy or safety. C, to determine patient adherence to prescribed therapies. Or D, to examine the role of biomarkers in drug efficacy. So, drug-being interaction between two drugs, the interaction between two drugs, and whether this interaction will affect the efficacy and or the safety. And what applies to this definition is B, as B is the correct answer to test drug combinations for potential reaction changes in efficacy or safety. Explanation: drug-being interaction trials focus on studying how two or more drugs interact when co-administered, altering efficacy or safety through potential pharmacokinetic or pharmacodynamic changes. Question 2. Which scenario best demonstrates safety monitoring in drug-being interaction trials? A, tracking INR levels in patients taking warfarin with antifungals. B, using pill counts to confirm participant compliance. C, comparing absorption levels during a drug and high-fat diet trial. Or D, analyzing demographic trends in trial populations. Best scenario, best example to demonstrate the safety monitoring in drug-being interactions or drug-drug interaction trials is what is applicable here, and the most accurate one is A, as tracking the INR levels in patients taking warfarin with antifungals. As warfarin, antifungal, both drugs, so drug-being interaction, and to check whether this interaction with affects the efficacy and or safety. So A is the answer, as safety monitoring involves tracking adverse effects. AEs like changes in laboratory values, for instance INR levels, to ensure risks such as increased bleeding with warfarin and antifungals are identified and managed. Question 3. What is the key advantage of compliance tracking in interaction trials? A, it ensures participants strictly follow regulated data transfer protocols. B, it prevents errors in pharmacokinetic parameter calculations like Tmax. C, it confirms patient adherence, reducing confounding factors in the results. Or D, it simplifies graphing of adverse event incidence trends. So the key advantage here, the main advantage of compliance tracking, so it's about compliance in interaction trials, would be C, as C is the more suitable answer. It confirms patient adherence, reducing confounding factors in the results. So C is the answer, as compliance tracking ensures participants adhere to the therapy regimen, preventing non-adherence from impacting the interpretation of the drug's pharmacodynamics or pharmacokinetics. Question 4. Which of the following is an effective tool for monitoring drug-non-being interaction in a trial? A, biomarker analysis systems to measure ALT and AST levels. B, CTCAE coding of drug adverse reactions. C, regression analysis to assess drug concentrations. Or D, risk management dashboards for all safety alerts. So it is A, as biomarker analysis systems to measure ALT and AST levels. is the applicable and most correct answer. So A is the answer. Biomarkers like ALT and AST are useful in identifying hematotoxicity during interaction trials. Hepato, liver toxicity, hepatotoxicity during interaction trials. Other tools listed do not directly measure biological effects of food or drug interactions. Question 5. When a trial studying polypharmacy shows increased renal impairment in elderly patients, what is an appropriate monitoring response? A. Disqualify elderly participants from future trials. B. Use EDC systems to flag elevated creatinine or reduced EGFR levels, um, glomerular filtration rate levels. C. Rely on quarterly adverse event analysis to determine trends. Or D. Reduce site monitoring visits to lower costs. So what applies here, as for example, or scenario, especially for trial studying polypharmacy, shows increased renal impairment, kidney impairment in elderly patients, and what applies to the kidney functions is here in B as creatinine levels and EGFR. So B is the answer. Use EDC, electronic data capture systems, to flag elevated creatinine and reduced EGFR levels. So B is the answer, as detecting elevated creatinine or reduced EGFR levels through EDC systems allows a timely identification and management of renal impairment risks, kidney impairment risks, ensuring participant safety during polypharmacy trials. And here we come to end of this lecture. Thank you very much and goodbye.